Development of zygote in flowering plants, germination of seeds, and adaptive features in developing animals. A comprehensive exploration of reproductive development in plants and adaptive features in animals, covering fertilization processes, seed formation, germination conditions, and reproductive adaptations across species. Lesson Overview 1. Plant fertilization and zygote development. The journey from pollination to zygote formation in flowering plants. 2. Seed and fruit formation. How embryos develop and differentiate into specialized structures. 3. Seed germination. Types of germination and necessary conditions for successful growth. 4. Adaptive features in animal development. Specialized structures in eggs and mammalian embryos. 5. Reproductive strategies. Comparing oviparity, viviparity, and ovoviviparity across species. Fertilization and zygote development in flowering plants. The journey from pollination to fertilization involves a fascinating series of events within the flower, transforming it into a seed-bearing fruit. Pollen grain germination. Upon landing on the stigma, the pollen grain absorbs a sugary liquid, swells, and begins to germinate. Pollen tube growth. The outer coat of the pollen grain splits, and a pollen tube emerges, growing down through the style towards the ovule. Male gamete formation. Inside the growing pollen tube, the pollen grain nucleus divides to form a tube nucleus and a generative nucleus, which differentiates into two male gametes. Gamete release and fertilization. The pollen tube penetrates the embryo sac, releasing the two male nuclei, which are then ready to participate in the fertilization process. Double fertilization, a unique plant process. First fertilization. One male nucleus fuses with the ovule to form a zygote, which develops into the embryo. Second fertilization. The second male nucleus fuses with the secondary nucleus to form the endosperm nucleus. Endosperm development. The endosperm nucleus produces the endosperm, which serves as the food storehouse for the developing embryo. Double fertilization is a distinctive feature of flowering plants that ensures both embryo development and nutrient provision through the endosperm. From zygote to embryo, development process. Zygote formation. The fertilized egg cell, or zygote, is formed after the fusion of male and female gametes within the ovule. Cell division. The zygote rapidly divides by mitosis, producing numerous cells that begin to specialize and differentiate. Embryo organization. These cells become organized into a structured embryo, featuring specialized tissues and rudimentary plant parts. Fruit and seed development. As the embryo matures, the surrounding ovary and ovules undergo changes, developing into a fruit containing seeds. The transformation from a single-celled zygote to a complex embryo involves precise cellular differentiation and organization into specialized structures, culminating in the formation of seeds within a fruit. Structure of a seed, nature's survival package. A seed is defined as the ripened, fertilized, and developed ovule, perfectly designed to protect the embryo and provide nutrients for initial growth. Key structural components include seed coat, the tough outer protective membrane that shields the delicate inner structures from environmental damage and desiccation. Hilum, a prominent scar on the seed surface, marking the point where it was once attached to the parent plant's ovary or stalk. Micropyle, a minute pore or opening, often located near the hilum, which facilitates the absorption of water and oxygen necessary for germination. Embryo, the innermost vital part of the seed containing the rudimentary future plant, complete with a root, radicle, shoot, plumule, and seed leaves, cotyledons. Seeds are broadly classified as monocotyledons, possessing one seed leaf, e.g., maize, rice, wheat, or dicotyledons, characterized by two seed leaves, e.g., mango, beans. The embryo, future plant in miniature. Plumule, the embryonic shoot that will develop into the stem and leaves of the new plant. Radicle, the embryonic root that will grow downward into the soil to anchor the plant and absorb water and nutrients. Cotyledons, seed leaf or leaves that store food in dicots or transfer food from endosperm in monocots. Endosperm, food storage tissue, may be absent in some seeds where food is stored in cotyledons. The embryo contains all the essential parts needed for a new plant to develop, arranged in a compact, protected package. Seed germination, awakening of new life. Germination is the process where a dormant embryo within a seed develops into a seedling. Before this vital process, seeds often undergo a rest period known as dormancy, ensuring optimal conditions for growth. Two main types of germination illustrate different developmental pathways. Epigeal germination. The cotyledons are carried above the soil surface by the elongation of the hypocotyl, typical in many dicots like beans. Hypogeal germination. The cotyledons remain below the soil surface as the epicotyl elongates, commonly seen in monocots like maize and peas. This coordinated process transforms a protected seed into an actively growing young plant. Types of germination: epigeal versus hypogeal. Epigeal germination. In epigeal germination, common in dicotyledonous plants, the hypocotyl elongates and forms a hook. The hook pulls the cotyledons above the soil surface. Cotyledons may turn green and photosynthesize. Examples: beans, sunflower, cotton. Hypogeal germination. In hypogeal germination, common in monocotyledonous plants, the epicotyl elongates instead of the hypocotyl. Cotyledons remain below the soil surface. Food reserves in the cotyledons are transferred to growing regions. Examples: maize, rice, wheat, peas. Conditions necessary for seed germination. Water/moisture. Activates enzymes, softens the seed coat, and facilitates the essential chemical reactions for growth. Oxygen. Required for cellular respiration to generate the energy needed for the seed's growth and development. Optimum temperature. Different plant species require specific temperature ranges for optimal enzyme activity and metabolic processes. Enzymes. Speed up the breakdown of stored food reserves, releasing the energy necessary for the seedling's initial growth. Food reserves. Stored in cotyledons, dicots, or endosperm, monocots, to nourish the embryo until it can begin photosynthesis. Seed viability. Seeds must be alive and capable of germination. Their viability can decrease over time and with improper storage. Adaptive features in developing animals. From eggs to embryos, specialized structures for survival. All vertebrates except mammals are egg-laying animals. The structure of eggs and embryonic development show remarkable adaptations that ensure survival and proper development of offspring across different environments and reproductive strategies. Structure of bird eggs, a perfect protective package. Shell. The hard, porous outer layer providing protection and allowing gas exchange. Membranes. Inner and outer layers just inside the shell, offering defense against bacteria. Albumin. Egg white. Rich in protein, it cushions the embryo and provides essential nutrients. Yolk. The primary food source, packed with fats, vitamins, and minerals for the developing embryo. Chalaza. Twisted cord-like structures that anchor the yolk in the center of the albumin. The bird egg represents one of nature's most sophisticated protective and nutritive structures, perfectly designed to support embryonic development outside the mother's body, while providing all necessary resources. Features of eggs across vertebrate groups. Fish eggs. Usually small with minimal protective covering, often transparent, and laid in large numbers. They typically have a jelly-like coating and are fertilized externally in water. Amphibian eggs. Surrounded by jelly-like substance that swells in water, providing protection and preventing desiccation. Usually laid in clusters or strings in water. Reptile eggs. Leathery, flexible shell with internal membranes that allow gas exchange while preventing water loss. Adapted for development on land. Bird eggs. Hard, calcified shell with complex internal structure, including air chamber, membranes, albumin, and yolk. Highly specialized for terrestrial development. The evolution of egg structures reflects adaptations to different environments and reproductive strategies across vertebrate groups. Mammalian embryonic development, the placenta. Uterus and implantation. After fertilization, the embryo travels to the uterus, where it implants into the uterine wall. This specialized environment supports the early stages of embryonic development. Nutrient and oxygen transfer. The placenta acts as a vital bridge, facilitating the transfer of essential oxygen, water and nutrients from the mother's bloodstream directly to the developing embryo. Waste removal, equally crucial, the placenta efficiently removes metabolic waste products from the embryo, transferring them back into the mother's bloodstream for excretion, maintaining a healthy fetal environment. Hormone production. Beyond exchange, the placenta is an endocrine organ, producing various hormones vital for maintaining pregnancy and helping the mother's body adapt to the physiological demands of gestation. The placenta is a remarkable temporary organ that forms during pregnancy, allowing for nutrient exchange while keeping maternal and fetal blood separate. Supporting structures in mammalian development. Umbilical cord. This vital connection carries blood between the fetus and the placenta, containing arteries and veins. Notably, maternal and fetal bloodstreams remain separated and do not mix. Amnion. The innermost membrane surrounding the embryo, filled with amniotic fluid. It acts as a crucial shock absorber and helps regulate temperature changes, protecting the developing embryo. Chorion. As the outermost membrane, the chorion facilitates oxygen absorption for the embryo and plays a key role in the formation of the placenta. Allantois. This membrane assists in respiration and excretion processes for the developing embryo. Over time, it integrates to form a part of the umbilical cord. These embryonic membranes and structures provide essential protection, nutrition, and waste removal for the developing mammalian embryo. Reproductive strategies. Oviparity. Oviparity is a reproductive process where embryos complete their development and hatch from eggs laid into the external environment. The laid eggs contain all the necessary nutrients for embryonic development, and the development occurs entirely outside the mother's body. Protective coverings vary greatly among species. Oviparous animals, such as most fishes, amphibians, all birds, and most reptiles, often produce large numbers of offspring. They have evolved diverse strategies like hidden nesting sites or parental guarding to protect their externally developing eggs. Reproductive strategies. Viviparity. Viviparity definition. A process of reproduction, in which the young develops and is nourished inside the female's body through the placenta and is born alive when fully developed. Characteristics. Direct nutrient transfer from mother to embryo. Internal development with maternal protection. Typically produces fewer offspring. Higher survival rate of offspring. Examples. A few fishes, like some sharks, some reptiles, and all mammals except monotremes. Viviparity represents an evolutionary strategy that maximizes offspring survival through extended maternal protection and nutrition. Reproductive strategies. Ovoviviparity. Internal egg development. Ovoviviparity is a reproductive process where eggs are retained within the mother's body, allowing embryos to complete development internally, without a placental connection. Yolk-dependent nourishment. Embryos rely solely on the yolk contained within the egg for all their nutritional needs, rather than drawing nutrients directly from the mother. Maternal protection. While development occurs internally, the mother provides protection and maintains optimal environmental conditions for the developing young until they are ready to hatch. Live birth of developed young. When fully developed, the young hatch from the eggs while still inside the mother and are then released alive. This strategy is seen in some sharks, snakes, guppies, and seahorses. Ovoviviparity represents an intermediate reproductive strategy that combines elements of both oviparity, egg-laying, and viviparity. Live birth with placental nourishment. Comparing reproductive strategies. Take a minute to observe the table, comparing reproductive strategies between oviparity, ovoviviparity, and viviparity strategies. These reproductive strategies represent evolutionary adaptations to different environmental pressures and survival strategies. Each offers distinct advantages and challenges for species reproduction and survival. Key takeaways. Plant reproduction. Flowering plants undergo double fertilization, a unique process that forms both an embryo and nutritive endosperm. The zygote develops into an embryo with specialized structures, plumule, radicle, cotyledons, that will become the future plant. Seed germination. Seeds require specific conditions to germinate, including water, oxygen, suitable temperature, and viable enzymes. Germination may be epigeal, cotyledons emerge above ground, or hypogeal, cotyledons remain below ground. Animal development. Vertebrates have evolved diverse reproductive strategies, such as oviparity, egg-laying, viviparity, live birth with placental nourishment, and ovoviviparity, internal egg development. These strategies involve specialized structures, like protective egg membranes, or placentas, to support embryonic development. These reproductive and developmental processes showcase the remarkable adaptations that have evolved to ensure the survival and success of plant and animal species across diverse environments.